Good evening, sir. Am I audible, sir?
Yeah, audible.
Thank you, sir. Thank you very much, sir. A warm welcome and very good evening to all dear participants. Resource person of today's session, Dr. Neeraj Kumar Agrawal, sir, and chairperson of today's session, Dr. R.J. Yadav, sir. Now, I would like to introduce today's resource person, guest speaker of the session, Dr. Neeraj Kumar Agrawal, sir. Sir is currently working as associate professor in the Department of Pharmacology, Government Medical College, Ratlam, and he is also member secretary in Institutional Ethics Committee. Sir had conducted more than 300 clinical trials, phase 1 and bioavailability and bioequivalence studies as investigator in pharmaceuticals. Sir had published more than 25 original research papers in various peer-reviewed national and international journals. Sir is the former principal investigator in Jubilant Life Sciences, a reputed pharmaceutical company for CT phase 1 and BNB studies. Now, I would like to request sir to please lead today's session on fundamentals of chromatographic techniques.
Yeah, yeah. Thank you, Amit, for introduce me to the audience. So, good evening, all of you. Yesterday we discussed about the pharmaceutical chemistry in reference to the drug development. So, in the pharmaceutical chemistry, we learned some about the techniques. Some terms were used like chromatography. Okay. So today we are discussing about the fundamentals of chromatography technique. So what is chromatography? So the chromatography, as name suggests, it's derived from the Greek word chroma and graphy. The chroma meaning color, okay, and the graphy means separation. So just it can be defined like the separation of the mixture. No. Okay. So separation of the compound in the mixture. Okay. Mainly color compound. But that is the old definitions because old definition is there. Uh, Nowadays we can also extract or separate out the colorless compound. Okay. So this is the definition as per the name chromatography. So chromatography provides a way by unknown compounds and separate mixtures. So any mixtures we have discussed about some plants extract, crude extracts are there. There are number of compounds are there, though they can be polar, non-polar. They might be low molecular weight or high molecular weight. Okay, there might be difference in the size. Okay, and there is affinity is also differ to the particular receptors like that. Okay, so this is the main thing you can just a second. So we can identify the unknown compound and then we can separate out by the chromatography procedure. Okay. So the chromatography procedure is a part of the analytical chemistry. Okay. So it is a part of analytical chemistry. You are just analyzing something to extract out. Okay. So analytical chemistry is a branch of the chemistry that provided the methods for identification of the substances, determination of the structure, and analysis of the composition of the matter. It's widely applicable in the various disciplines of chemistry such as inorganic, organic, physical, and biochemistry. So this chromatography, as far as our medical curriculum is concerned. So chromatography basically it comes within the biochemistry. Okay. So we are dealing with the chemistry part with the biological. Okay. So if you want to do something in the chromatography, So you must have some knowledge about the analytical chemistry. Okay. Before this, it's not possible that you will clearly get the idea how the which type of chemical substances are separated out from the chromatography. So you must have knowledge about the biological science and the chemistry. Okay. So analytical methods are also applicable in the fields of environmental science, agricultural science, biomedical, clinical chemistry, forensic, and the space research. Okay. So because of it is widely wide applications of the chromatography. So you can see here there are different type of field that where we can use the chromatography. Okay. So definitely our medical fields, as we have yesterday we have discussed about, there is a very good role of R&D, research and development, the drug development. So in this category, we provide an introduction in different topics of analytical chemistry, or we will be dealing with the types of the chromatography. So techniques. Okay, try to I will try to introduce about the chromatography briefly, and because of brief is that because it's a very huge topic. Okay, so you can read number of pages, okay, if you want, but as far as the medical field is concerned or R&D is concerned, research and development, so I will briefly introduce this topic, and you will easily get the knowledge. Ultimately you will after this session you will get what is the chromatography and where we can use it. So chromatography is a technique for separating mixtures into their component in order to analyze. First of all, you have to analyze, okay, a small molecule, large molecule, what is present in that particular mixture. Okay, then identify, analyze, identify, and then purify, and then quantify. Okay, so separate out, then fill, quantify. So all are interrelated to this chromatography. Okay, so types of so there is types of chromatography. Okay, on the basis of these principles, there are types of chromatography. The first is column chromatography. Okay, so there are whenever you see the standard books or papers, research papers, so you will get a number of classifications of chromatography. Okay, I'll try. I am trying my best to easily you will get a simple idea and easy classification of the chromatography. Okay, so you just go with me. Start with there are three types of chromatography. The first is column chromatography. Okay, second is paper chromatography, and third is thin layer chromatography. Okay, so I'm just repeating again because of this chapter is totally dependent on these types. The type one is column chromatography. Second is paper chromatography, and third one is thin layer chromatography. Okay. So first of all, we will discuss the column chromatography. Okay. The rest of two, the paper chromatography and TLC, thin layer chromatography. Okay. The principle of rest of two, paper and TLC, both have same principle. Okay. So we will discuss in our later part. Okay. So first of all, we will concentrate on the column chromatography. Okay. So, as far as the chromatography is concerned, you have to remember some important part of chromatography. There are two phases of stationary phase and mobile phase. Okay. So always the chromatography, the basics of chromatography. So the main principle which mainly depends on the two phases. The first is stationary phases and second is mobile phases. Okay. So whenever you extract something, so there are two phases. The one is stationary, the second is stationary. So first of all, in column chromatography, you can see the stationary phase is column. Okay. So you can easily remember by this, the stationary phase is the same which is name suggests, like column chromatography, the stationary phase is column. Okay. paper chromatography, which is stationary phase is paper. Okay. And thin layer chromatography. Okay. So you can see there are thin layer of silica. So this is the stationary phase. And generally mobile always mixture. Okay. Mixture to be separated. In the paper chromatography, mixture of amino acids and solvent, like that. Okay, that is the same. So we will just whenever you I will give you some idea, then you will get about what is the meaning of these stationary phase and the mobile phase. Okay, so column chromatography is based on, so first, okay, we will now focus on the first type of chromatography, that is called column chromatography. So column chromatography is based on mainly three things. Okay, this is important. If you know this, then you will easily get the idea of chromatography. The first is size of the molecule. Okay. First is size. What is the size? Size means molecular weight. So size or molecular weights are both are same in the biochemistry. Okay. As far as the biochemistry is concerned, so we generally assume the size means it's like a molecular weight. So different type sizes or molecular weight of the compounds are present in the mixture. Then we have to separate it out. So for the size of the molecule, we generally use the gel filtration chromatography. Okay. For the size of the molecule, we generally use the gel filtration chromatography. Now come to the second, charge. So what type of charge are present on the molecule? Okay. Means anion, cation. So there is a mixture of compounds, and some compounds are anionic, some compound have anionic charge, or some has cationic charge. So for separate it out, we use the ion exchange chromatography. Okay, ion exchange chromatography. Now third part. Third part is depend on the affinity. What is affinity? Affinity to the particular molecule. So we just add some part like some coenzymes which has there are different type of affinities are present of different affinity of the compound present in the mixture. Okay, so that particular affinity to that particular uh, where we just add them. Okay, so particular affinity that they will bind to the particular specific substances, and ultimately we can the that particular enzyme can be separated out. Okay, any amino acid. Okay, so by this is called affinity chromatography. First is size, gel filtration chromatography. Second is charge, on the basis of charge, ion exchange chromatography. And third is affinity chromatography. Okay, so now we will discuss one by one. So you just see this. This is the table which is showing the first is gel filtration chromatography. This gel filtration chromatography, also called size exclusion chromatography. Okay, it is also called size exclusion chromatography. So here is there are two type of mixture. There are mixture of two proteins. Okay, suppose we are taking the mixture of two proteins, and we have analyzed, then we get that there are different sizes of molecular weights of proteins present in the mixture. Okay, so what will we do? We just use gel, that is the most common used gel is Cefadex. It is called Cefadex dextran. It's a mucopolysaccharide which is used in column. Okay, like in like test tube in column. Okay, so we see there, and then we pour out that particular mixture in that gel. So we will see there is large molecule eluted first and small molecule eluted later. So what is the meaning of this? So you can see by just I am you can see in this diagram, you can see here. So there are gel molecule is there. Okay, gel molecule is there. This is gel molecule. Okay, and this is a mixture. This mixture contain large protein as well as small protein. This is large protein, small protein. In this gel, there are small holes are there. In a small hole, only small protein can enter, then exit then again enter, then exit, enter, exit. Okay, okay. And the large protein will not be able to enter into the small pores of this gel. Okay, so it directly goes into the lower side. Okay. So, okay, so which type of protein will reach early at bottom? Definitely large protein. Okay, large protein, because of it is not able to enter into the small hole which is present on the gel particle. Okay, so ultimately large protein will go first and a small protein will come later. So large protein eluted first. This is the term used in the chromatography. This is called eluted first. Eluted means which comes first. Eluted first. There is illusion. So this large protein eluted first and small protein eluted last. So now by this method we can, you can see here, okay, you can see here the small protein, it's small protein, large protein, okay. So this large protein, okay, large protein eluted first. You can see here. So it is showing we can separate out with the gel gel chromatography. So gel filtration chromatography, this is called size exclusion chromatography. The application of this is, the application of size is give great advantage in the separation of a purification of, where it is used, sugar, polypeptides, different sizes, viruses, proteins, enzymes, hormones, antibodies, and nucleic acids, DNA, RNA, etc. The main application of SEC is a gel filtration or size exclusion chromatography, is to separate or analysis of protein and other water soluble polymer from their mixtures. It's also used to examine the stability and properties of natural organic matter present in the water. So different types, and you can see in this. Okay. So now come to the second one. Second is called ion exchange chromatography. So ion exchange chromatography. So there is mainly, I have earlier said that there is mixture of two proteins which analyze charge positive or charge negative, anionic or cationic. Okay. So we have, first of all we have analyzed ki, okay, there are some cationic molecule, there are anionic. Then we will okay, we will then plan ki we will do the ion exchange chromatography. So what we do here there, so there are different type of methods are there. So first common method we I am discussing here. So you can see we will take the uh, uh, carboxy methyl cellulose, which is present on the any test tube. Okay. So then we will pour that mixture. We will pour that mixture. So carboxy methyl cellulose, which has the negatively charged. Okay. It is a negatively charged solvent. This is which is negatively charged. When we pour the mixture, so you can see, so what would happen? So positively charged will attract to the carboxy methyl cellulose and negative charge eluted first. Okay, so negative charge molecule eluted first because of, definitely there would be attraction of the opposite molecule, and positive charge molecule separated. Then positive molecule remain in the bind with the carboxy methyl cellulose. Then we will just add the buffer and then we can separate it out. So by this method, we can separate it, the uh, charge positive or negative. Okay. So in the diagram, you can see. So this is the diagram which is showing like that. You can see here. So this is the mixture is present on this beaker. This is the column. In column, you can see carboxy methyl cellulose solutions is there. Now you will pour it. After pour it, you can see here there are positive charge protein bind to negative charge beads. Okay, positively charged proteins and that bind to negative charge, and negative charge proteins flow through. So this is negative charge eluted first. Okay, so by this method you can just separate it out. So what is the application of ion exchange chromatography? So you can see in medicine, it is used for the analysis of sugar and amino acids. Definitely you can see, you must know this amino acid, and different type of amino acids are there. It's also used for the separation and purification of blood components. Okay. It's used for the analysis of impurities or quality control in the chemical industries. In agriculture, it's used for the analysis of micronutrients in the soil samples. In the petrochemical chemistry industry, it's used for determination of sulfur compound. Okay. And in the environmental chemistry, used for detection and separation of toxic pollutant present in an aquatic and atmospheric environment. So definitely, as far as the medicine is concerned, you can just see there is separation purification of blood component. Okay. So now move to the third important is affinity chromatography. So in the affinity chromatography, mainly it depends on the affinity of particular compound. So there are a mixture of two proteins. One is enzyme. Okay, just one I'm taking one example. Like one is enzyme. Okay. So what we do? We do be mixture of that particular mixture, then we pour out into the one column which has already present coenzyme, or that coenzyme has the affinity to that particular enzyme. Okay, so what would happen? The enzyme will attract with the coenzyme, and the rest of the molecule, other molecule will eluted first. Okay, so other molecule will eluted first, and that particular coenzyme will remain with the enzyme. So same things happen in chromatography. So you can see with the diagram. So this is affinity chromatography. You can see here the first is this is analyte, this is mixture, and this is ligand. Ligand which has the affinity to particular compound which is present on this mixture. Now what would happen? This analyte retained in the stationary phase, liquid, and the this it has the affinity. This blue one has the affinity to this ligand which is present on the solvent. This is solvent. Okay, but the remaining part, remaining mixture is eluted from the sample. Okay. You can see here this is desired protein. This desired protein which is which which has the binding material. Okay, so it attaches this black one. Okay, on the rest of molecule which eluted first. Okay, so this is the affinity chromatography. And what is the application of affinity chromatography? So affinity chromatography offers high selectivity. Very important because of it has the high selectivity. Okay. So you can see resolutions and capacity in most protein purification. So protein purification because of specificity is very much important in the case of biology, like antigen antibodies and the other amino acids. Okay. So most protein purification is done by the affinity chromatography. It has the advantage of utilizing a protein's biological structures or functions for purifications. As a result, purification that would otherwise be time consuming and complicated can often be easily achieved with affinity chromatography. Because of affinity, you must know the enzymes. They have specific enzymes present for the antigen antibodies and the amino acids and biological structures. They have specific, which are affinity to the specific substances. So we can utilize this thing. So affinity chromatography is very much important in the case of research and development of human research and development for the drug, especially in the human environment. Okay. So now we will move on the principle of paper. Okay. So now we have finished the column chromatography. Now we will focus on the rest two like paper and TLC. And I have discussed earlier that both the principle of paper chromatography or thin layer chromatographies both are same. So what is the difference between mainly difference between paper chromatography or TLC? So paper is a old version. Okay, I have mentioned in my slide earlier that paper is a old version and TLC is a new version like that. Okay, so paper is definitely TLC. It's a new version. So TLC is very costly. Okay, but definitely it's very effective. You know, it mainly used in the R&D nowadays. TLC, thin layer chromatography, HPLC, you have definitely heard HPLC, high pressure TLC, HPLCs are there to extract the components. So this is very important nowadays. So you must know the principles of paper and TLC. So for this, I am just giving one examples. Okay. So just we take a one paper, just on one paper, we take the three amino acids. The name is A, B, C. Okay. Then we take a beaker. In a beaker there is solution that is called methyl cellulose media. Okay, so first of all we take a paper, then we add in the bottom side we add the A, B, C. There are three amino acid. Okay, then we take a beaker. In beaker there are media, that is solvent, that is called methyl cellulose media. And this methyl cellulose media is a non-polar. Okay, it's a non-polar. So we just dip the paper in the solvent. Okay, we just dip the paper in the solvent. Then solvent will move upward because the capillary actions are there. So that particular solvent will move upward, and by the movement of the solvent, it tried to carry amino acid with it. Okay, so the solvent will try to carry amino acids with it. Okay, so in this case you can see the mobile phase is mixture of amino acid with the solvent. Okay, because the amino acids is moving as well as solvent is moving. Okay, after wetting whole paper, whenever whole paper become wet, okay, then we take out the paper, and then we do the stain with the anhydrous solution. Okay, so it produces a purple stain, particularly to amino acids. Okay, so then we see the location of the particular amino acid A, B, C, where they are located on that particular paper. Okay, so just you can see the diagram. I am just showing you. This is the diagram. You can see this is the solvent. This is the solvent, carboxymethyl cellulose. This is the non-polar, and this is the paper. Okay, this paper is dip into this. There are three amino acids like blue, purple, and red. A, B, C. You can see here. Now, okay, now you can see there is capillary action. The solvent will move upward. So you can see some remain this side, some more up this side, and then you can see this is the diagram. You can just see 2 centimeter. The red amino acid only reaches to 2 centimeter. Then this 6 centimeter, and this 8 centimeter, and last solvent will reach into the. Okay, this is the total 10 centimeter solvent reach there. Okay, and start from origin. Origin is there. So how you interpreted this? Most important thing is the interpretation of the paper chromatography. Okay, or thin layer chromatography. The principles are same. So this is you can see in this principle you must remember two important things. Polar compound will be soluble in polar solvent. This is the general, you must know this thing. The polar compound will be soluble in polar solvent, and non-polar compound will be soluble in the non-polar solvent. Okay, what would happen? So early state, what is showing the early state? Okay, so you know the solvent is non-polar. Okay, so non-polar compound will take the non-polar easily. Okay, okay, and the polar compound will not go, only that cover the short distance. Okay, like this. Just see this. So early state, am I not, like just I am again showing the, again showing this diagram. You can see this. This is non-polar. Okay, so whenever non-polar compound go, go upward, so the non-polar compound will reach, will will cover more distance. Okay, so you can see the blue is non-polar. Okay, and red is polar. Okay, more polar. You can say more polar. Okay, so purple is more polar than blue. Okay, red is more polar than purple. Okay, or you can see purple is more non-polar than red, or blue is most unpolar, like that. So this is the interpretation. Okay, and if solvent is, if solvent is non-polar, okay, then early, okay, early stain. This is called stains. Early stains, middle stain, the late stain. Okay, so early stain always opposite to the solvent. You can say, you can say like that. Early stain always opposite to the solvent. Oh, okay, suppose this solvent is non-polar. Okay, it can be polar on the basis of what we need, what are our objectives, then we can use the solvent. Different type of solvent can be taken for the different type of mixture. So I hope you got my point. So early stain amino acid polarity is opposite to the solvent polarity. Okay, and late stain amino acid polarity is similar to the solvent polarity. This is the general principle of both thin layer and TLC. So in TLC technique, the adsorbent is coated on a glass plate. Just I discussed on a glass plate which acts as a stationary phase. Usually we used alumina or silica gel with little calcium sulfate for the production of thin layer chromatography plate. So first of all, we make a plate where solvent is there. The solvent or mobile phase which we use to thin layer chromatography is very important steps. Okay, it depends on mixture, what type of mixture we need. So we on the basis of this we use the solvent. Compared to other chromatographic technique, it's applicable for quantitative analysis of the sample. So this is again important nowadays. Mainly, it is used for the quantitative analysis of the. This is the thin layer. It's thin layer chromatography. Again, TLC plate is there like that. This is the advanced version. Okay, so you can see the same process. Process is same. A, B, C. Same TLC chromatograph. A is early stain. Then C is late stain. So definitely what solvent? If I say A is polar, so definitely the solvent will be non-polar. So this is the just you can see the interpretation by this. What is the application of thin layer? So it's very useful for the analysis of a large number of compounds that are not volatile or too labile for gas other chromatography because of there are different types of drawbacks of other chromatography. So hence it is mainly used for compound that is non-volatile too labile for other chromatography, and it is also used in the forensic science chemist. It's applicable for the analysis of amino acids, plasticizers, antioxidants, ink dyes. It has extensive application in inorganic analysis and a new method. This is called IF, just high pressure TLC. This is called HPLC. HPTLC is a short by HPLC. It's a thin layer chromatography which is used widely in the pharmaceutical analysis. Yesterday I have discovered their HPTLC which is required for the pharmaceuticals, pharmaceutical chemistry, pharmaceutical analysis. You have to use the HPLC along with the other techniques like NMR, electrophoresis like that. So thank you. Now session 4.
Thank you, sir. Thank you very much. I hope that all participants get benefit from today's session. And now this is the query session. I request to all participants if there is any query regarding today's session, then please raise your hand or write your query in the chat box. Dr. Anupama, I sent you the unmute request. Please unmute yourself and ask your query.
Hello, sir. Sir, when can we use the gas chromatography technique, GC?
Sorry, I didn't understand.
When to use GC, gas chromatography?
Chromatography.
Yes, sir.
So actually, these all are basically actually gas liquid chromatography and liquid chromatography. You can see at the different places. Okay, I have just mainly concentrate on the classification of the chromatography which deals with the medical science. Okay, so gas chromatography mainly which is used in the environmental science and the in the inorganic chemistry. Okay. So basically, as far as the medical science is concerned, we will mainly focus on these. Okay. this is as per my opinion. Don't go more detail because of what I have told you about the chromatography. It is more than enough for the R&D in the pharmaceuticals or especially in the medical field. I think Dr. Amit, I think Amit, because of it's a theoretical topic, I think there,
Right, sir. Right, sir. Right, sir.
yeah,
There is a
yeah,
query from Dr. Rajni Thakur. He written his query in chat box. Dr. Rajni
yeah.
Thakur, I sent you the unmute request. Please unmute yourself. Yes,
No,
yes.
sir. I had no query, but I have got the study material, so I thank you in that message box.
Okay,
Sorry.
okay. Thank you, ma'am. Thank you. I got your
I
point.
have
Uh,
got the
uh,
study
you can just
material
share this
in this tab.
Definitely,
PPT
definitely.
to the
So I
audience. Yeah,
thank
yeah.
you.
Definitely, I requested to all participants that the
Okay.
study material will be shared on the Statisco app and access will be provided throughout the year. So mostly tomorrow I will share all the PPTs as soon as we get from the our guest speakers. So I hope that our
Yes, sir.
queries will be answered. If there is any query remaining, then I request to all participants, then they can mail us. I will provide mail ID in the chat box. And thank you, Dr. Neeraj Agrawal sir, for a very informative and knowledgeable session. I hope that all participants will get benefited from today's session. Now, I would like to introduce today's chairperson, Dr. R.J. Yadav sir. Dr. R.J. Yadav sir is currently working as a senior consultant at National Institute of Biologicals, Ministry of Health and Family Welfare, Government of India. Before that, sir had been retired as a Director Grade Scientist G from National Institute of Medical Statistics, NIMS, ICMR, New Delhi. And Dr. R.J. Yadav sir is having 31 years of research experience, 20 years of experience in administration in ICMR, New Delhi. Sir is having the experience of evaluation of 30 studies for CARE, WHO, UNICEF, USAID, Department of WCD, Government of India, etc. Sir guided and explained PhD and MSc students of many universities. Sir has published 98 research papers in various international and national journals and also published 90 scientific reports. Dr. R.J. Yadav has received various awards such as Professor P.V. Sukhatme Award in 1999, WHO Fellowship by Government of India in 1998 and 2005, and Fellow of Indian Society of Medical Statistics in 2007. Sir has been member of various scientific societies like General Secretary of ISMS from 2011 to 13 and currently life member of 6 international and 10 national scientific societies. He is also member of SSC of NIN. Now, I would like to request Dr. R.J. Adar sir to please share his views and conclude today's session. Please, sir.
Hello,
Yes, sir. Yes, sir.
good evening, everybody.
Very good evening, sir.
Good evening, sir.
Thank you, Dr. Agrawal, for nice talking. This was a very informative and very encouraging. This lecture was. So, hello.
Yes, sir. Yes, sir.
Yes,
Audible.
sir, audible, audible.
Okay, so I think in this contest, if this any study relating to Indian system of medicine and homeopathy, nowadays it is called Ayush, then it will be a great pleasure to speaking on this platform, and we will be collaborating any study relating to this in future. So I think with this, I can say this is the great success for this conference, this meeting. Thank you all. Good evening.
Thank you,
Yes, sir,
sir.
thank you.
Thank you. Thank you. Thank you, sir, for nice words. And definitely, we are working on it. And this is the first program in the Ayurveda field, collectively combining the Ayurveda, stats and biostats together, sir?
Surely, because we have worked a lot in this for the Ministry of Health and Family Welfare and Department of Ayush also. That time it was a department, now it is the Ministry of Ayush. Okay, so looking forward to be contact for this any research projects in the future.
Yes, sir, definitely, sir. Thank you very much for your presence,
Thank,
sir.
thank you everybody. And
Now, from the Joint State Training Center of Rajasthan, we convey our sincere thanks to all the participants for their active participation and commitment for today's session. We heartily thank the guest speaker of the today's session, Dr. Neeraj Agrawal, sir, who kept participants engaged by the way of very informative and interactive discussion. We also thanks for sharing today's session, Dr. R.J. Yadav, sir, and sharing his experience and views about the session with the participant. Now, this is the end of today's session. We again thanks to all of you. There is a one information that regarding the notes of sessions. The recorded session will be shared weekly on Monday through our statistical app. If there is any query regarding access, if you found any query, then you may call at any time uh, in the morning from 10 to 5. Otherwise, you can directly access. I have sent the instructions in the WhatsApp group. You can follow them, enter the batch code, and easily access that notes and recorded session. Currently, we have shared one book of Fundamental of Stats and Research Methodology. And tomorrow, maybe I will share all the PPTs I have get by today. Thank you again. Thank you very much.